Okay. Good evening, everyone. Let's get started. Okay. Welcome to week 11 of this course, Bioinformatics, Algorithms and Applications. So this is the second last week. So this week and the 12th week will be mostly talking about applications, right? So applications and different kind of statistical techniques that is used to analyze biological data. Now. The thing is, in the course, the data that is shown is mostly protein data, PDB database. So data has been downloaded from PDB databases and other sources, but mostly protein data. I will give some more examples regarding how different types of algorithms is used in different biological data, because again, bioinformatics is a vast field and like it is growing, and in the past 10 years it has grown so much, and different types of data have been generated. We are analyzing those kind of data, and so on and so forth. So it will be I just wanted to give you a little bit more exposure to different kinds of work and research that is going on, and so forth. There was one Autodoc tutorial which I am not going to cover because it's a tutorial and it has already been covered. If you have any issues with Autodoc, you can let me know. Probably tomorrow we can have a small tutorial on that if there are any issues, or we'll skip that. Other than that, yeah, so there's the awk programming part of this week as well. So we are going to discuss some of the commands, and basically like we do that, some questions might come from, like, basically in your exams, from that part some commands may come, and like you might be asked to write or choose the answer with respect to how the output will be, and so on. Okay, so the main focus of today's session would be to see different types of methods, different types of algorithms, specifically a little bit of machine learning algorithms and how they are used. And in the end, we're going to discuss some relevant research that has also gone on regarding those, and also the kind of work that I do. I am more familiar with. I'm going to discuss it a little bit more. Okay, so let's get started. Okay, so in terms of, so we are starting with the applications of the algorithms, and yeah, different types of applications of bioinformatic algorithms, right? Okay, so say for example you have your data set. Now you want to do some analysis on that data set, be it whatever kind of data. It can be protein data, it can be some other types of data. You have a data set and you want to do some analysis on that. So there are some checklists first to be maintained, right? So the first and foremost question to be answered is, what is your research question? What do you want to find from the data? That question will lead you to the different types of analysis that you can do on the data to get that answer, right? So the first and the primary step is identifying the underlying research problem to be solved. So the first important task is to identify the research question. Your question should be clear. Your question should be could be broken down into smaller set of questions. You might want to answer one of those questions sequentially, right? So the asking the question is the most important part. What exactly you want to find from the data, right? Now, from the different types of algorithms that we have learned about in this course, including the initial alignment algorithms and different types of evolutionary algorithms that can tell you about how much divergence have gone on, and so on and so forth. So all of these actually tell you something about the data itself, right? So if you're doing multiple sequence alignment with multiple individuals or multiple species of a particular gene or a particular protein, and you see some divergence in terms of the alignment, you see some amino acids have been changed, some particular type of amino acids have been replaced with another type of amino acid, and so on and so forth. So that would tell you about how the evolution has occurred, how has the species diverged from each other in terms of their their genetic structure or their protein architecture, right? So for all of these, you need to have basically a well-defined problem, and which will guide the selection of the subsequent data and features. So it should not be the other way around, that you have the data, now you are trying to find something in the data. It can be done. It's called an exploratory analysis. But if you have a particular question in mind, the data also should be conforming to that question as well. Right. Now, majorly the data that we have and the kind of problems that we face in bioinformatics, or the kind of problems that we want to solve in bioinformatics, is basically of two categories. One is a classification, and one is some sort of value prediction, right? So both of these things, so classification would be to predict a binary outcome, either a 0 or a 1. So it can be status of a disease, if the person has a disease or does not have, or it can be something like presence or absence of an amino acids, or the presence or absence of a regulator or something like that. So it's a binary outcome, either you have it or you don't have it. So that's that kind of data is used to do classification analysis, right? And in terms of real value prediction, it's mostly the data will be quantitative continuous data. Say for example, you want to predict the hydrophobicity of a particular amino acid. You have all the other information regarding the amino acid and other amino acids. From that data, you want to predict what is going to be the hydrophobicity of this amino acid. So it is going to be a continuous variable. It is not going to be a binary outcome if in terms of if it's hydrophobic or not. It will be a continuous variable and a value that will tell you if the amino acid is hydrophobic or not. So the classification is based on discrete categories or yes/no outcomes, and the real values are basically you are predicting exact numerical values on continuous or quantitative data. So the binary predictions are done through algorithms which are mostly classification based algorithms, and in terms of predicting real values, it's a framework we call as regression, right? So yeah, these are some examples of different types of questions that you could answer with these set of either through classification or through regression. So for example, you want to predict a structure of a length of a peptide, and you want to predict if the structure will be an alpha helix or a beta sheet, or some coil or something else, right? So you have data of other proteins, you have their sequence, you know which peptides are making alpha helices and beta sheets or coils or whatever. You have all the data together. Now you have your protein structure or protein sequence. You want to predict what is going to be the structure of a particular area of that sequence. Is it going to be an alpha helix or a beta sheet or something else, right? Yeah. So that would be one such example. You could predict binding sites with respect to DNA, RNA, or other ligands, or some other signaling molecules, or something like that. You could also predict in terms of if a particular mutation in the genome or in the protein can cause some disease or not. So one good example of that would be to see you take a set of variants present in all individuals in your data set. Say for example, you have 10,000 individuals in your data set. You have the mutation information from all those 10,000 individuals, and you know 2,000 of these are having a particular disease. Now you would want to make a classification model or a logistic regression model based on all the features. The features would be your mutation data, and you would try to predict the disease outcome of the individual as a binary variable 0 or 1. So analysis like this could be done using classification algorithms, and also some other types of analysis. For example, if you want to find if a residue is buried or exposed, or if it is contributing to the solvent accessibility or so and so on. So yeah, so it can be it must be a 0, 1 or a binary outcome of the data you are trying to predict. The features of the data might be continuous or might be binary, depending on the different data that you have. There is no restriction on the features that you are using to predict the outcome, but the outcome definitely needs to be a binary variable, not a continuous state, right? that would be the classification types. Next would be different types of regression. Say for example, you want to uh, predict predict a mutation change, like if there is a mutation, what will be the change in the protein stability, right? Or if you have data between interactions between two different pairwise amino acids across all possible combinations, what will be the interaction between two different proteins that you have. So these kind of things, these kind of continuous variable predictions are done through regressions. Okay, so yeah, so it is the data set is strictly dependent on the quality of the training. Your model, what you are building, would be definitely very much indebted to the data that you are using. So it should be from high quality databases like PDB, Uniprot, so on and so forth. And the most important thing is basically your data should not have redundancy. So when you're training a model, if you have redundant data in your model, it will skew your model, it will hamper the prediction capability of your model to predict a particular outcome. So it should not be repetitive, the data should not be redundant, and if it is redundant, it would overfit. What is overfitting and underfitting, we're going to come later in the slides, but yeah. And basically, if you're using protein data, if you want to remove all the different redundant protein sequences from the database, you can use some alignment algorithm like BLAST or cluster to get a particular alignment score, and if your certain sequences go above a certain alignment score, you're going to remove those. And diversity is important to capture the biological variations, right? So there are certain terms that you should be aware about in terms of the data and the models itself, right? So first would be the training set. So you are using a part of your data to train your model, and then you are trying to predict some outcome from that model, right? So the main set or the training set is basically the initial features and parameters of the algorithm the model would be trained on, and the test set would be the rest of the data that you are wanting to validate your performance based on this model. So generally, if in machine learning algorithms, there's a 80-20 split. So you take the whole data set, you split it into 80% and 20%. Now the 80% would be used to train the data, and the 20% of it would be used for prediction of the data and for validation, right? So this is not your actual actual prediction you're doing. You're just training the model. This is this training phase. In the training phase, you need to train and test the model, right? So for the training, you'll use a significant amount of the data, and you're going to validate or predict or test your model on the remaining small amount of data that is remaining, right? Now, it can be a completely independent set that the model has never seen to ensure an unbiased final evolution, right? So if your training and test set kind of overlaps, it does not kind of make the model more robust. It brings in more bias to the model, and if you want to predict something else maybe later with respect to protein structure or whatever you are trying to predict, it might not predict well for the other data, because your model has been trained on and tested on the same data, so it devalues the model quality, right? Now your data will have a lot of features. What are these features? Generally in a tabular data, we consider the columns to be distinct categories that we have values for, and we consider those as features. Now features can be anything depending on your data. So if you're having a protein data, your features could be amino acid sequence, the molecular weight of it, of the protein, the conservation scores across different sites. You could have structural features in terms of if that particular region is forming a secondary structure, what kind of secondary structure it is forming, what are the different long range and short range contacts and residue interactions, and so on and so forth. So the feature can be anything that is describing your independent variables. So your dependent variable. So your dependent variable in this case is basically if you are wanting to predict if a particular protein will bind to another protein or not. So that is your dependent variable, and your independent variables are all your features here, which would be amino acid composition, molecular weight, conservation score, secondary structure, and other things that you could think of in terms of whatever data you have. So this slide is to give you a general idea that in when you are making a model, these are the terms that you should be aware of. There should be a training set on which you are going to train the model. There should be a test set which is part of the whole data, but you have divided the data into a larger training and a smaller test set, and you are going to test your model on the test set. Now ideally that test set should not overlap with your training set. That way the model will be more robust and will have more unbiased evaluation. But if there are overlaps, it will be a little bit more overfitting to the data itself, right? And then you have different features of the data. The features are basically whatever data is describing your dependent variable. In this case, probably your dependent variable is something of related to the protein structure or binding or affinity or something like that. And what are the different characteristics of the protein or the amino acid composition of the protein. So those will be your features. Right? Okay. So yeah, this is a representation of that. So this is your whole data set. You split your data set. Mostly it's an 80-20 split. So you use this data set to you use the training set to train the model, and use the testing set to validate and test the model, right? So in this case, many a times they do cave cross-fold validation, right? So basically, it is the training set will be used to train the model. The validation set will be used to assess the performance of the model, and the k-fold cross-validation will be, you divide your training set into k small sets, and you use each of those to predict, and then see the mean error or the mean squared error, whatever cost function or loss function that you are using, you try to assess that. Now in a lot of cases, what can happen is you will have the data, but not exactly in binarized form, right? So for example, if one of your feature of the data is basically the secondary structures, right? Yeah, if one of your features of the data is secondary structures, now in the secondary structure column you either have helix or sheet or coil written for all the proteins that you are analyzing. Now the idea would be that the model cannot interpret these English terms or these technical terms. What it can interpret is a binary variable, which is a 0, 1 or 2, right? So in this case we have 3 categories. So what we do is basically we divide this should be one column. So in one column you have helix, sheet, coil, and so on and so forth. So you divide this one column into 3 different columns. So if it's a helix, it will be a 1, then the other 2 columns will be 0, 0. If it's a sheet, the middle one will be 1, the other 2 columns will be 0, and if it's a coil, the last one will be 1 and the rest will be 0, right? So this is what is known as one hot encoding. So you convert your categorical variables into binary variables across different number of data points, depending on the total number of categories that you have. So if you have 3 categories, you will have 3 columns for this. If you have 10 categories, probably 10 or 9. So there is also the concept of so since we are going to use a regression like framework, there is also the concept of multicollinearity. So if your data has a lot of features which are correlated with each other, then your model will overfit and the performance of the model will not be very good, right? So it in most cases, if there is multicollinearity, it cannot estimate the beta, which is the effect size of each of the features. So in those cases, your model will not work well, right? So now comes the concept of the underfitting and the overfitting of the data. So the idea is that failing to learn the underlying relationship between the variables is called underfitting, whereas learning the noise in the training data is called overfitting. We are going to in a more formal way define underfitting and overfitting. This is just to give you an idea. So the idea is that if your model is so if your data is so this is a distribution of your data, right? So this is a distribution of your data. If your model is a straight line like this, your model is not explaining your data in no way whatsoever. So this is an underfit model where your model is not being able to fit to the data. In this case, you see this is a parabolic curve, right? So if a parabola is fitted here, this will be a good fit. That parabola can and fit the data well, right? But if you see there are no, it's not a, it's all of the data points are not falling on the line, right? So there are some haphazard data points here and there. Now if your model is fitting to each and every data point carefully, then your model is fit to your data completely, right? It is called overfitting, that it has fitted to your data completely. It has not learned the pattern. Instead what it has learned is the noise with the data, because the data points that are deviating from the original line has some noise component to the data as well, right? So what it is doing is it is not learning the general pattern, which will be a parabolic fit. Instead what it is learning is the noisier patterns, and so it is fitting to each and every data point in the data, which is not good. So these are the three cases mostly that you are going to come across. So this is the underfitting, where the model is nowhere close to describing the data. The good fit would be the ideal model which is fitting to the data but not overfitting to each and every point. And the overfit would be your model has completely transformed itself to fit in the noise from the data as well, which is not good at all, right? Yeah, so underfitting can be caused by a model without sufficient complexity to describe the signal, and overfitting can be caused by a model with too many parameters or by continuous training after it has learned the true relationship between the variables. So the idea is that you can train it multiple times. Iteration wise, you can split the test and the training data differently in different iterations and train your model on those iterations. So what can happen is, if you do it multiple times, the model will learn the noise from the data as well, and it is going to fit itself to the noise as well, which is not good for the model, right? It is going to be predicting well for that particular data set, but it will not be able to predict for any data that it has not seen before. So that is an issue with overfitting, right? We are not going to go into the learning rates and all, but again, this is a very interesting take on that. Okay. Yeah, so coming to a little bit of details of overfitting and underfitting. So this is your cost function, right? I hope all of you are aware what a cost function is. So the idea is that this is your model, this is your actual data, and this is your predicted data from the model. So x given d is basically your predicted data, and y star is your actual data. So what you do is basically subtract from the predicted data and square it. So this is basically the loss function. This is the loss calculated from the data. So for example, let us take this example. So your line is passing through like this, but your data sets are not all data points are not actually falling on the line, all of them, right? So what you're doing is you're trying to minimize the distance between the line and the data points. So how do you do that? You evaluate a cost function. The cost function would be to check the distance between the line and the data points for each data point. So it'll check, and it'll give a squared value, because it can be negative or positive, that does not matter. We are just want to check the physical distance, right? Now it will check the distance between the data points and the curve, and it will square it, and that will be your cost function, which you are going to try to minimize, because you want your line to be as closely fit to the data as possible, not overfit, but as closely fit to the data as possible, and not the noise. So what you're going to do is, through every iteration, you're going to try to minimize this cost function, which is generally a difference in squares, right? It is called as mean squared error, MSE. So this error can be quantified in terms of noise, the bias of the model, and the variance of the model. So mathematically, what is happening is, if you are overfitting the model, your variance is going to be too high, right? So this is the second case where you're overfitting your model. So in this case, the variability, you see the data points, the variability is too high, and what you're fitting is nth powers of x. So you're going to very high powers of x to explain the model itself. So that is, it is introducing the variability in the in the data, and that variability is causing the data set to overfit, right? But in case of underfitting, what is happening is the bias is too high. The initial conditions, the initial bias of the model is too high, so that it cannot fit to the data at all. So your, what you need to do is you need to have a choice of model that balances between the bias and variance. So this is a plot that you'll come across a lot in if you do if you learn if you study statistics and basically bias variance modeling and so on and so forth. So the idea is that there is one point in terms of the error. If you calculate the error at the lowest point of the error, it can happen is where your bias and variance are both lowest. So that would be your optimal model, where you have a good balance between bias and variance, so that your data does not underfit, neither does it overfit. So the key takeaway from this is, if you have high variability in your data, the model is going to overfit. If you have high bias in your data, if you do not have any much variability, it's kind of similar, then it is going to underfit. And if your variability is high, then it is going to overfit. And there is one point where basically you have the lowest variance and the lowest bias, and in terms of the error, it is close to the lowest. So that would be your optimal model to predict your outcomes, right? Okay, now we are going to talk a little bit about the traditional machine learning methods. We're going to cover the basic machine learning methods in as simple way as possible. I'm going to try to give you an intuitive understanding of what each of these things do. We are not going to implement, we are not going to implement any of these right now, but like just to give you an idea about what each of these things do, right? Okay. Regression and SVM we are going to talk about in details in the further slides. We are going to see some examples as well. But we are going, let us come to gradient boosting first. So gradient boosting, what it does is, it is using an ensemble of weight prediction models, typically some decision tree models, to make predictions. And it can make predictions like an active drug can be predicted from molecular descriptors such as molecular weight and the presence of particular chemical compounds. Individual predictors are combined in a stage-wise manner to make the final prediction. So what you are doing is, you are dividing your model into smaller models, and you are taking each of these model and doing a prediction, and you are trying to combine all those predictions into one, and basically trying to predict for the final data. So this is an ensemble of smaller models which can predict the local architecture but cannot learn the whole data. So what you are doing is, you are using multiple models for multiple sections of the data, and then you are using the predictions of all of these, combining all of the prediction of these models to give you an outcome, right? That would be gradient boosting. Principal component analysis, on the other hand, is basically a very well-known technique of dimensional reduction. So it uses eigenvectors. So basically PC1 and PC2 are eigenvectors of the data, which are orthogonal in nature, capturing the most variability, the most variance, you can say. So PCA finds a series of feature combinations that best describe the data while being orthogonal to each other. It is commonly used for dimensional reduction. In the case of height and weight of a person, the first two principal components correspond to a linear combination of height and weight, describes positive correlation, where PC2 might describe other variables that do not correlate strongly with those, such as percentage of body fat or muscle mass, and so on and so forth. So the idea is that you have, as we were talking about different features, right? So in one data set you can have 10,000 features of describing one amino acid or one protein, right? So now 10,000 features or 10,000 values for one data point, it is very hard to visualize, right? But what we do is, if there is so high dimensional data, what we try to do is reduce the dimension and project the variance of the whole data into 2 dimensions, which is actually principal component analysis, and these two dimensions are orthogonal to each other. So whatever you see in terms of PCA for the first two dimensions kind of holds for the entire data for, say for example, 10,000 features. So generally it is used as a dimensional reduction technique, but it can also be used for linear combination of traits or something else as well, right? And finally, clustering is basically something that we have probably touched upon a little bit earlier, is basically grouping similar things together. So in this case, what they are doing is grouping the cells by their gene 1 expression and gene 2 expression, right? So you have three distinct clusters here, where in this particular cluster you have both high gene 1 and gene 2 expression. In this cluster you have cells with low gene 2 expression but high gene 1, low gene 1 and high gene 2, and in this case it's low gene 2 and high gene 1. So these are done, the clustering is done based on gene expression of two genes across different types of cells. So what you're doing is you're bringing similar type of data together and clustering them together, saying that these fall under the same category, right? So these are all general machine learning methods, and we're going to talk about regression and SVM in a little bit more detail. Now OLS is basically part of regression. Ordinary least square is a statistical method that is used to estimate the parameters of linear regression model, and it is also known as the best fit line. So the idea is that you have a dependent variable and an independent variable. You draw a scatter plot between the x and y axis, and you try to find the best fit line across that in the whole data. And to do that, ordinary least square is used, right? So the idea behind that is, again, as I was saying, there needs to be a cost function or a loss function which you are trying to minimize, right? So this approach focuses on minimizing the sum of squares of the residuals, which reflect differences between the observed values in the dataset and the predicted by the model. And in linear regression, the relationship of the dependent variable yi and a set of independent variable is given like this. So this is the linear regression model. So your yi will be a vector of all your dependent variables, xi will be a vector of all your independent variables, and you're going to fit this model and estimate alpha and beta from the model. How do you estimate alpha and beta? You use OLS, right? So the coefficient beta represents the parameter of regression, which I often term as effect size, or you might have heard telling me effect size and so on and so forth. So effect size is actually the beta of the regression model. So the coefficient represents the parameter of regression and summarizes the influence of each input on the dependent variable. So the idea is that, say for example, for one mutation you got a negative beta while predicting protein binding affinity. So the idea is that if you have that particular variation in the gene, in the protein, your binding will go down by whatever beta has been reported. So the negative beta meaning it is negatively associated, positive beta meaning it is positively associated. You can think about it as a scaling factor from the independent variable to the dependent variable, right? So the beta will tell you the strength of the association, the strength of the correlation, not exactly correlation but association, I might say. Okay. Now the beta is the slope in a straight line. If you consider this as an equation of the straight line, the beta is going to be the slope of the straight line, and alpha is the intercept, right? So the term alpha is the intercept and captures the baseline value of yi when xi values are 0. So basically if you put xi equal to 0, beta will be equal to alpha. So beta is the value of the alpha will be equal to yi, right? So the alpha value will be the value of the dependent variable when the independent variable value is 0. That is going to be the bias, or this is known as the bias alpha, and also the intercept of the model, right? And yeah, so the sum of square residuals, which explicitly is the target of OLS, is given by this particular expression, which is, so basically what you're doing is you're so this is your original variable, original value of the thing that you're trying to predict, and this is your model. So you just have done yi minus alpha plus beta xi whole square, and then you square it. So basically this is what linear least square is. So the least square approach chooses alpha and beta to minimize the residual sum of square. So what you're trying to do is basically minimize the sum of square from this sum of square. This is the sum of square across all data points. So yeah, you're minimizing the sum of squares with respect to alpha and beta, and the use of the squared error is an analytical convergence because you can have negative values as well. So if you're adding up the errors across all data points, so if you have negative values and positive values, some of them can cancel out, which will be an underestimate of the total error in the data. So for that reason, mostly the squared term is used. And this so if you do a differentiation with respect to this and try to find the minima of beta, the expression that comes out is something like this. So you can directly estimate beta if you have x bar is the mean of x, y bar is the mean of y, and xi, yi are different data points. So if you have all the data points, you can directly find beta using this particular term. You do not need to fit the model. Okay, so yeah, so this is a schematic of the regression model that we are trying to show. So these are the yellow ones are your data points, and this is the model that has been fit, this straight line. What you are trying, how you are going to evaluate the model is basically how far the particular data points are from the actual model. So in terms of that information, you're going to quantify. So what can happen is in some cases it can have a negative difference. So if you add the negative and the positive difference, it will cancel out and it will not tell you the exact information about the model itself. So what we do is we use squared terms for each of these residuals, and then we try to minimize the residuals, right? So yeah. Now some major advantage of least square method is to relate it to its flexibility, interpretability, speed, and explanatory power. Specifically because of the expected linear relationship between the response and the independent variables, one can immediately infer the effect of changing a variable value on the prediction, right? So further, the underlying statistics enable calculating confidence intervals both on the predictions themselves as well as the parameter values. So what you can have is, so this is your beta, right? So with the beta you can have some standard error with respect to the beta, and also you can have a standard error with respect to the predicted dependent variable as well. So you can have some error associated with it, which your model will tell you that with how much confidence it is telling you that the value of your predicted variable will be this. There will be always some error associated with it, always some noise associated with it. So that is what is getting calculated with respect to OLS, right? So yeah, so because of expected linear relationship between the response and the independent variables, one can immediately infer the effect of changing a variable value on prediction. Further, the underlying statistics enable calculating confidence intervals on both the predictions themselves as well as the parameter values. A key approach for estimating uncertainty in parameter estimation is bootstrapping. That you do it for 100 iterations, keeping one random data point out, and you see how much standard error are you getting, how much variability are you getting. So that would be a good idea to do with help of bootstrapping. So bootstrapping resamples the given data with replacement to create a new sample data set of the same size. Then parameters are re-estimated using the sample and compared against the original parameter estimates by creating a distribution of the desired statistic. So basically how much deviation is there with respect to your parameter if you do the iteration 10, 20, 50 times. So that is going to tell you the standard error or the variability in terms of the parameter. So bootstrapping is a good way to do. Jackknife is one such method that we have discussed also last class. So that is also something that you might want to do. Okay, now just give me one moment. I'll just be back. Okay. Okay. One second. Okay. So one example of implementation of OLS and regression was this particular paper. So here what they did was they used multiple linear regression. So for multiple linear regression, you so here in linear regression only x is your independent variable and y is your dependent variable. So you're trying to predict a one single dependent variable from one single independent variable. What can happen is you can have multiple such independent variables, right? So if you have like 50 features in your data, you would want to capture almost all 50 of those to be independent to be predicting for your dependent variable. So if you have multiple predictors, multiple independent variables, then it becomes multiple linear regression, right? So these people used a multiple linear regression framework underlying with a Bayesian framework, which basically you have a prior distribution. So you have a distribution from which you can pick the regression parameters. So you know that this is the range of the regression parameters, and this is a distribution from which you can sample your parameters of regression, right? It was used to model similarity between ecoregions as predicted by geographical distance and environmental conditions. And ecoregions are large cohesive areas of land or water that are often described in terms of species, their ecological dynamics or environmental conditions. In this paper, what they used was a Jaccard dissimilarity index. So Jaccard index is a very simple index. You can have a Google search quickly. It's basically if you have two set of groups involving some parameters. So Jaccard index will be defined as A intersection B by A union B. So how much is common between the two by the total union of the two. That is Jaccard index, right? So they use this Jaccard index to capture differences between ecoregions, and this index was used as a response variable in the models, and they tested whether the distance between ecoregions is explained either by abiotic or biotic factors. So basically, I don't know if I have the paper open. Yeah, this is the paper. So they have taken all the biodiversity data and all the climate data and so on and so forth. Here they have seen the correlations of the different data, and here is basically the regression. The regression that they are doing with respect to the Jaccard index, right? So, and here they are mapping out how much are similar, how much are dissimilar across different animals and plants in different geographical locations. You can read this paper. This is a very, very interesting and very nice paper. If you want to see how regression works in real world data, this paper is a very good example of that. Okay, so this was one example. The next example was this particular paper, and in this study the authors used linear regression models to compare estimates of phylogenetic divergence times between taxa as estimated by simple or complex models of molecular evolution. So they have already categorized different phylogenetic distances or divergence times, and using different molecular evolution models. So what they tried to do was use linear regression to estimate the congruence between these different types of models in terms of their predictions. So what they did here is they built different types of molecular evolution models, and each model gave a prediction, right? So what they wanted to see was they used linear regression to see how similar or dissimilar the predictions between two different models were. So that this is used regression on models, not on directly the data itself. So this is the paper. You can take a look at this paper as well. This is also there. So these are your different models. So GTR plus this is one, and JC is one, and they have used, yeah, they've used these set of regression models across uh, different models to see which one are actually congruent in nature. So these here they have compared different models across different species. So this is also an interesting read if you want to take a look at. Okay, so yeah, so I'm going to quickly give you an idea. So implementing linear regression in Python is quite a simple thing to do. There are dedicated Python libraries for implementing machine learning models like linear regression. So if you want to run an OLS using Python, you can use this particular library called the stats models, and you can store your training and test data in pandas data frame, and this is a pseudocode for this. So basically this will be your training data, X will be your training data, Y will be your test data. So your model will be defined by this is your model, stats model.ols, Y is your test, X is your training, and then you fit the model using model.fit, and then you make predictions. So this is a small pseudocode that can be used to run a regression model on a data which is stored as a pandas library. So yeah, in Python it is not very difficult to implement models like this. The more difficult part would be to get the data of your choice, clean the data, and make the data ready for regression on machine learning models. That would be a more difficult task, right, in real life. Okay, so we talked about regressions.
Sir,
Yeah,
I have a question.
yeah, yeah.
I'm not familiar with Python or any kind of programming language as I'm in just graduation level.
Okay.
So do we have to like focus it
No, no, no,
for
no,
now,
no,
or
no, no,
we
no, no. This
can
is not part of your course.
learn this?
This week, tomorrow, and the next week. I'm going to divert a little bit away from the course because why I'm going to do that, I'm going to tell you, is because the examples and all of the things that has been taught in the course, because the course is old, there has been a lot of advances in different levels in bioinformatics from then to now. So I'm going to try to give you a little bit more real life examples of the research that is going on currently, and also that can be done in the future, rather than the kind of analysis that has been done in the past, and so on and so forth. So across these 3 sessions I'm going to tell you a lot of things which are not going to be part of your examination. So you can only focus on the assignment problems and what has been taught to you in your actual classes. uh, this session, tomorrow's session will be mostly focusing on the topics that has been taught, but also the next session, week 12, I'm going to focus a little bit on the real life applications of it, and basically how you can use it. You can, since you're doing an NPTEL course like this, the best way to learn would be to do something hands-on with some data at some level. So I'm just giving you some resources, some ideas on how to get the data and analyze data, right? So I'm just telling you that it is not very difficult to analyze, and this kind of questions will not come for Python and R, and those kind of questions will not come. I'm just giving you an idea about how things are done in real life.
Okay, so thank you so much, sir, for giving us the real life updates.
Yeah, yeah. Okay. Okay, so yeah, this was just to show that it is, for regression is not a big deal. Regression, you can do it in like 5 lines of code. So, but as I was saying, the more important thing would be to get the correct type of data and to clean that data, because biological data has a lot of noise, a lot of redundancies, a lot of like bad data. The idea is that, like, machine learning model will not take much time to run. The main time take that will take is to form the research question and to get the data and to clean up the data. These 3 things are the major things. If once you get the data, you have a lot of models at your disposal with pre-built libraries like stats models, TensorFlow for deep learning, and other Python libraries, or there are libraries in R as well, and in other languages as well, which you can directly use to do this modeling. It's not very difficult. Okay. Okay, so another type of machine learning model that is used is SVM, support vector machines, and these are a set of supervised learning methods. Okay, so I think I have not covered what is supervised versus unsupervised learning. So supervised learning is where you have labels for the data, right? So you have categories for the data, and you're trying to put your data into one of those categories, right? So that's supervised learning, where you know what categories are there. In unsupervised learning, regression would be an unsupervised learning. Is like when you're trying to predict, you have no control over the data, over the prediction that you are going to do. It's the model, how it is learning and how it is predicting. There are no categorizations that the model has to fill in. So if you are using 5 categories, if you have 5 categories in your data, and you have one data point, you want to fit that data point into one of those 5 categories, that would be a supervised learning. And if you want to see if this is forming a new, if this is not falling in the 5 categories, this is a new category in itself or something else like that, that's an unsupervised learning, that you do not have prior knowledge about the data or the classification of the data, right? So SVM is a supervised learning method where you have some labels or some idea about the data itself, and it can be used in terms of image classification, text classification, and other bioinformatic routines. So image classification is something, and image data is something that I don't think has been mentioned much in the course, but I want to tell you that right now, and also like in the next 5-10 years also, from the past 2-3 years, image data is one of the major building blocks for machine learning and deep learning models across bioinformatics. Any kind of image data, be it MRI data, it can be X-ray data, it can be some other kind of image data, it can be just pathology. So for example, I'll tell you one small example is, if you want to predict, so you have, say for example, from a hospital you have collected a lot of images of tumors, of particular tumors, and you want to predict if those tumors are benign or metastatic in nature, or like benign or cancerous in nature, right? Based on the pathology, a radiologist or a pathologist will tell you, after checking the cross section of the tissue, if the tumor is benign or not, right? Same thing, if you want to do it with in a machine learning model, you need to classify that data previously. So some training data will be there where you are classifying each of the data to be part of a tumor or not, part of a benign tumor or not. So if you have a data set like that, and you are given an image of a patient, and you are asked to tell that does this patient have a benign tumor or a cancerous tumor, using the model, using the image data, you can predict that if the person is having that or not. So image data currently is a very big deal right now. Image processing and computer vision is a very big deal, specifically in bioinformatics and biotechnology, and in bioinformatics is image data is very useful, because the computer can learn a lot from the image data. The machine learning models can learn a lot from the image data, specifically MRI, specifically USGs or X-rays or things like that. The algorithms are very well trained to learn data types like that. So SVMs are types of models that are used for classification of these type of data, right? So so it is used for classification, but can also be adapted for regression tasks, but like it's not a good idea to do regression with SVMs. Okay, so what SVM does internally is basically it checks similarity between any two pairs of data points, right? So based on the concept of similarity, SVMs use nonlinear kernel functions. Do not worry too much about the mathematical terms. I'll not go into much details, but I'm just wanting to give you a whole picture. So SVMs use nonlinear kernel functions that is used to transform the data from a higher dimension to a lower dimensional set, which enables linear expansion by finding optimal boundaries between two sets of data points. So it can be that forms decision boundary between classes and support vectors or the data points that lie close to the decision surface to maximize the margins between the classes. So this is how it looks like, right? Not to go into much of the mathematical details. So what it is trying to do is create, so this is your feature x and this is your feature y. What it is trying to create is a hyperplane between two sets of data points. So you have a group of data points here, you have another group of data points here. What the algorithm is trying to do is, can I have a partition between these two sets of data points so that these two can be shown as independent clusters. So in this case, yes, so this is the hyperplane. So this is an imaginary plane between these two set of data points that this algorithm will find, right? So again, why are we talking about planes and not straight lines? Because we are talking about high dimensional data. So we are talking about data where you will have 10,000 features, right? It's not a two feature data, or it's not a one feature data, that you have 10,000 features. You are reducing the dimension from 10,000 to 5 or 6, and then you are trying to find dividing planes between these two sets of data, right? But this representation of the data is not actually the true data itself, right? Because it's a higher dimensional data. You have reduced it to two dimensional, and then you are trying to find a partition between these two, right? So that is what SVM does. It tries to find this particular hyperplane, and it finds the margin, right? It finds the margin of the hyperplane as well, like margins on which some data points might lie, or the closest data point to that to the last line will be the margin. So this is what SVM does. So again, ignore the mathematical expressions, but I'm going to keep these so that in the future, if you want to take a look at these PPTs will be available. You can just go and take a look if you are interested. So that is why I've kept. You do not need to focus much on the mathematical expressions, right? So these SVMs are primarily focused on determining the hyperplane, as I was saying, that optimally divides the data into particular classes based on the maximum margin. The hyperplane is defined such that the minimum distance between the data points in different groups is maximized, right? So the idea is that this to this distance should be maximized, this to this distance should be maximized. So inter group distance between the data points should be maximized is what SVM is doing, and this is basically the optimization cost function for that. I'm not going to go into details, and yeah, so the basic idea is that to form that hyperplane between two groups of data. So I'm going to discuss again an example of this SVM implementation. So this is the paper. I don't think this paper will be available. It is available to me because I am on the ISC VPN, but I don't think this is going to be available to you guys. Can you just take a look if full text is available for this paper for you guys or not? If not, I am going to put a PDF of this in the drive. Just check if it is opening or not.
No, sir, it's not available for us.
Okay. So I'll put a copy of this paper in the drive. Okay. Okay. So what this paper has done is basically they have taken they've taken data from healthy and unhealthy tomato plants, and they've taken photos of those tomato plants. Now they've used these photos to classify if a tomato plant is healthy or not. So for example, whether can this be implemented? So for a farmer, they can go and click a photo. If they are unsure if this particular plant is infected or not, they can click a photo, and if that photo is run through a model trained on the data like this, it can tell you directly based on the photo if the plant is infected or not infected. So what they did was they developed classifiers that could improve agricultural sector. So they collected images from existing databases containing images of healthy and unhealthy tomato leaves, and they conducted image pre-processing steps. I'm not going to go into details how they extract image data and all, and they used 60% of the data set for training and rest for observation and for model performance, and say they could determine, they could find a hyperplane and could determine. So just let me go here. So these are the kind of data that they have used, right? So all these mathematical things, I'm not going to go there. Yeah. So this is,
Sir, I guess
sorry.
I guess something similar was done using satellite photos of
Yeah, yeah, definitely, definitely. So these,
satellite photos of agricultural land,
definitely,
and
definitely, it can be done.
it would tell the infected area from the not
Definitely, yeah,
infected
yeah,
one.
yeah. So the thing is, so much of data is available right now because we have so much of sources of data, so much of image data, so much of text data also. If you are interested in doing things like these, the best thing would be to find a data set that can interest you, that is of your interest, and play around with it using different models, SVMs, regressions, whatever, basic machine learning models. You don't have to do deep learning on those kind of things. If you are interested, there are a lot of these data sets available. You can do a lot of predictions using this data. But again, your question must be good enough, and your data must be very relevant to your question. As I was saying in the initial slides, that the choosing of the data is very important. And the data should be like complete enough to answer your question. Okay, yeah, yeah, definitely. There have been multiple such projects. There have been a lot of projects in my institute as well, where they use MRI data to classify if a person is having cognitive impairment or not, does not have cognitive impairment, if the person will go have neurodegenerative disease in the future or not. So yeah, image data is very useful, and since we have a lot of image data available in public repositories currently, so it is very useful. Okay, anyway, so these guys used these three regression models, so these three machine learning models, and they showed that the SVM had the highest accuracy across all three. And they, yeah, sorry, yeah. So these are the kind of problems that can be solved using data like this and models like these. So yeah, I wanted to give you an example of that. Okay, so finally we are going to talk a little bit about artificial neural networks. So these are a little bit more complicated models than normal basic traditional machine learning models like regression or SVM or random forest, etc., etc. So what they do, the name comes from the fact that the form of the mathematical model is inspired by the connectivity and behavior of the neurons in the brain, and was originally designed to learn the function of the brain. So these models were not exactly used for prediction early days, because these were used to understand the brain itself, right? However, neural networks are in common use in data science, are obsolete as brain models, and now are just machine learning models that can offer state-of-the-art performance in certain applications. Interest in neural network models have grown exponentially in recent decades owing to rapid advances in architecture and training deep neural networks. So the basic idea between neural network is that they are universal function approximators. What does it mean? It means that with a very few set of assumptions, a correctly configured network, it can approximate any mathematical function to an arbitrary level of accuracy. Why is that required? Is I'm going to give you a little bit of context. Is, say for example, for SVM, we are using this particular cost function and we are trying to reduce this cost function, right? So in the back end, when you're running this model, so what the model is trying to do is trying to minimize this particular function. Now if you have multiple models in a single model, multiple models training on small snippets of your data, what can happen is converging all the models towards the same thing or towards a minima is going to be very difficult. For that, you need good approximations, good numerical understanding, good numerical methods to approximate these models to have some sort of conversions. So that is what neural networks do very, very, very well. So it can approximate any mathematical function to an arbitrary level of accuracy. And the idea is that if any process can be thought of as a function of a set of variables, then that process can be modeled to any arbitrary degree of accuracy governed by just the size or the complexity of the model. So basically, it will converge. The idea is that it will converge the model and will give you an approximate estimation of it. So again, now the universal approximation is not very rigorous from a mathematical standpoint, but it does highlight one reason why interest in neural networks has persisted for decades. But it does not guarantee or does not provide a way of finding optimal parameters. It is going to give you some parameter, but there is no guarantee that that will be the optimal one. It will find a minima, it will find probably a local minima, but it will probably not be able to find a global minima. So there are some trade-offs. If you are willing to do that, if your data is robust enough that you are willing to do this kind of trade-offs, then using neural networks is probably the best idea. So here a little bit of more information about neural networks I've provided. So these artificial neurons are what are known as the building blocks of neural networks, and these artificial neuron is basically a mathematical function that will convert an input to an output. It will map an input to an output, which is the function of a mathematical function, and that is what an artificial neuron is. A single artificial neuron takes any number of specific input values, applies a specific mathematical function to them, and returns an output value, right? And it is written like this. I'm not going to go into the mathematical interpretation of that, but so that is one function of one artificial neuron. When you want to make a neural network, then the artificial neurons has to be arranged in layers, with the output of one layer being the input of net. So I don't have a representation here. Artificial neural network. Yeah, sorry. Yeah, this is how it looks like. So the input will be coming from here. So these layer of neurons will take the input, provide some output to the next layer. So for the next layer, the input is coming from the previous layer. So the layers are nothing but mathematical functions. It is taking the data, applying a mathematical function to the data, and giving an output. So starts from here, outputs to the next layer, then outputs to the next layer, and finally, which is the output layer. So you can have layers of these artificial neurons for multiple of these mathematical functions, depending on how you want to predict from your data. So it gives you a lot of flexibility in terms of different mathematical functions, different types of shrinkages, different types of predictions, right? So it gives you a lot of flexibility. The nodes of a network can be thought of as the various can be thought of as holding the y values from the above equation, which becomes the x values for the next layer, which is basically it holds the predicted value from the previous layer and gives an output to the next layer, right? So so there are different types of architectures of these neural networks. So there is one very commonly known architecture is convolutional neural network, which is a very good classification model, and it has fully connected layers that are usually used to produce the final classification output. So CNNs are very well known models for classifications. So a lot of AlphaFold, actually, if you read the paper of AlphaFold classification, they have used a lot of classification models. And one more small thing that I want to highlight is AlphaFold. Probably you guys are familiar with, but one also paper that has come out recently, it's a 2026 paper in Nature. This is Alpha Genome. So here also they are using machine learning, deep learning models to predict the functional effect of a particular mutation, right? So the models are not described here. Here all the data is described. So I think if you go to supplementary information, so yeah. Mm, Not sure if they have given any figures for that. Let's see. No, these are not figures for the model. No, it doesn't look like they've given the model architecture here. Yeah. Yeah. Here is the model architecture, if you can see this. So this, what they're doing is, if there is a splice site mutation in the genome, is it having any effect on the actual splicing or the gene expression downstream? So they're taking the variant. So this will be one layer, this will be one layer of network, this will be another layer of network. So first it is going to predict whatever gene are relevant. In the next layer, it is going to predict if these are the genes, what are the gene expression differences, and in the first final prediction, it is going to aggregate and predict that. So this is for one such model. This is another model. This is one network. This is another network. This is another network. This is another network. So these are, so they have not put it in terms of the neural network, but these are the different predictions that are going on for each level of the neural networks. So it is doing both classification and regression. It is predicting some values, and it is also classifying if it is a good variant or the bad variant. So it is predicting if it is what is the effect of the variant, if it is a pathogenic or a likely pathogenic or whatever, right? So also it is predicting other parameters as well. This is a very interesting paper. This paper came out in this year, I think January, if I'm not wrong. So this is continuation with respect to the AlphaFold paper. So in the AlphaFold, they predicted effects of mutations in different secondary structures and tertiary structures of the proteins, and the final what is the structure of the protein going to be. And this is an extension of that. It is using that data and using different models of its own to predict if the mutation is there and if the structure is getting hampered, what is the downstream gene expression change that is going to happen. So again, if you guys are interested, please take a look at this paper as well. Okay, now we are going to talk about the parameters by which we evaluate the models. I'll quickly go through these. These are not very difficult to understand. So first is the accuracy. Accuracy is going to tell you number of correct predictions divided by the total number of predictions. How many correct predictions out of the total number is that you're doing. So ideally, your prediction accuracy should be high. Yeah, but the accuracy is good. It should be high, but it gives a false positive sense of achieving high accuracy. The problems arise due to the possibility of misclassification of minor classes samples being very high. So the idea is that if your accuracy is high, that is the meaning is that you're predicting, your correct predictions are high with respect to the total number of predictions, but it is not telling you about what predictions you're doing correct. As in, like, are you identifying the true positives and also the false negatives, or are you just identifying the true positives and not identifying the false negatives? So that information is not given by accuracy. So we have different metrics for other classifications as well. So precision is basically is going to give you an information about how you're doing with respect to the false positives as well. Are you are you being able to only identify is your model just giving true for all data points, that your true positive rate is high, your false positive rate is also high? ideally it would be your true positive rate is high and your false positive rate is low. That would be a good model, right? So that this expression is given by precision. For recall, it is the opposite. Is your true positive rate high, and also are you being able to identify the false negatives? The correct negative are also are you going to are you being able to identify? So that is given by recall, and F1 score is a derived metric from precision and recall, and this is the formula, and this is a harmonic mean between precision and recall. And so if you want to combine precision and recall into a single number, this is the one. So a high F1 score means the model performs well on both precision and recall. So ideally you do not calculate separately precision and recall, you just calculate the F1 score, and if your F1 score is high, your model is working well. The other parameters that is used to talk about the quality of the model is the area under the curve of the ROC curve itself. So generally useful for binary classification tasks. The AUC value represents the probability that the model will rank a randomly chosen positive example higher than a randomly chosen negative example. It is ranging from 0 to 1. With if your value is closer to 1, that means it is better. If it is closer to 0, it is worse. So here you define true positive rate, right? So it's similar to precision, which is true positive rate, true positive by by false positive. It is equal to recall, true positive by false negative. So this is false positive rate, this is true positive rate, and true negative rate as well. So this is not precision on recall. This is precision, and this is recall, right? So yeah. So true negative rate will tell you that out of actual negative cases, how many did the model correctly identify as negative. And for the true positive rate, it will tell you out of the actual positive cases, how many did the model identify correctly. So one for positive, one for negative, and false, same. So basically, how many are of them were misclassified as positive or negative will depend on the false positive rate and the false negative rate. Now this is what the ROC curve looks like, and AUC is basically area under the curve of the curve that you have for the ROC curve. So this what you are plotting is the false positive rate on the x-axis and the true positive rate on the y-axis. So ideally your model should be somewhere here close to 1, that it is the true positive rate is very high, close to 1, and the false positive rate is very low, close to 0. So if it is close to 1, it's a perfect model, but if it is close to 1, that is also possible that it is overfitting, right? Remember that the model can overfit. You have to be careful about that. If it is 0.5, the model performs no better than random guessing, and if it's less than 0.5, that it is not well good at all. So at least 0.5 and above, the ROC curves should have an area. So in this case, the ROC curve has an area of 0.79. This is 80%. This is not bad. It's decent. Right. Okay. One last thing to discuss here is a confusion matrix. Is something like a chi-square table, right? So it's predicted no, actual no, predicted yes, actual yes, and out of total number of numbers, what are the different distributions? So you can have true positive, which will be predicted yes and actual yes. This 100 is true positive. False positive would be actual no but predicted yes, so that would be 10. False negative would be predicted no, actual yes, which is 5 here, and true negative would be predicted no and also actual no, that would be 50 here. So this is what is termed as a confusion matrix. So generally in binary classification, this kind of matrix are used, and we do a chi-square estimate of this to statistically test if the differences are significant or not. So yeah, that's all from this side. One thing that I have not covered here is the awk commands, right? There's one question in the assignment about awk. We're going to come to that, but I am not going to discuss all the commands, but I found a very good tutorial if you guys are interested. So this is basically a tutorial. They have provided the files as well, uh, which you can use to practice different commands. So all the commands are given, what commands does what is given. So this looks like a very good tutorial that if you want to follow, this will take probably 30 to 40 minutes to follow this tutorial, and if you can follow this tutorial, you'll mostly know about awk, right? So again, yeah, if you have any doubts, please let me know. I'll cover it in either tomorrow or the next class. The things that I'm skipping, one is the awk programming, and the second is basically what else did I skip? Yeah, if yeah, the autodoc thing, the autodoc and the awk programming. If you have any issues, please let me know. I'll try to cover if required in the next sessions. Okay, now we are going to move into the assignment questions. Before that, if anyone has anything to ask. Okay, then we are going to move to the assignment problems. Okay. Yeah. Uh, the answer is B. Someone answered, I think, A. If I'm not, A is never going to be correct because maximum energy requirement is never the case for stability. Right. Whenever you are going to be stable in terms of the molecule, you should have lowest energy. So maximum energy is never going to be the correct answer. So yeah, the second one is the correct answer. So yeah, so maximum interactions and minimum energy of the complex. So yeah, maximum covalent interactions, if it is there, it's the best. Yeah, and most negative free binding energy, which will be the lowest one. Okay, a lot of B's. Yeah, that is correct. The purpose of docking is to find binding affinity and other things. Yeah, that's correct. on. Okay, couple of E's I can see. Okay, we have not discussed this in this session, but it tells you the current record number in the current file. So basically, tells you the file number record, which tracks the current line number being read in the current file. NR tracks the total line number being read across all input files combined, and NF is the number of fields in the current record. So FNR is file number of records. Yeah. Okay, that's correct. Sensitivity and specificity. So Pearson's correlation is just going to tell you, and only you can do for continuous data. Absolute error also you can do for continuous data. Sensitivity and specificity is related to precision and recall, and from that you can create the ROC curve and basically use the classification evaluation performance from AUC. So that's correct. Yeah. Okay, moving on. Okay. Yeah. Let me just show you the sensitivity, specificity, precision, recall. Okay, I think where's the formula? Let's try this. A is the correct option. I'm just trying to see if there are any. Uh,
I was confused. It was 1 minus something. I was
specific.
confused between A
Uh,
and D.
A is correct. I'm just trying to find if there
Mhm.
is anything. ROC curve. Yeah, true positive, false positive is given. Yeah. So false positive is 1 minus specificity and true positive is sensitivity. So it's the same thing. ROC is same as 1 minus specificity by sensitivity or false positive rate by true positive rate. So yeah, A is the correct option. Okay. Okay, no answers yet. Wait another minute. Yeah, that's correct. It's the same. So true negative rate is basically specificity. 1 minus specificity is false negative rate. So specificity will be true negative rate. It's the same. It's 0.75. So these kind of tricky questions can be asked in exams. Just be careful about the definitions and what each of these things mean, and I think you'll be all right. Okay. Okay, I think it'll take a little bit more time. Okay, wait another minute and then I'll discuss. Okay, the answers are correct. So what is given? You're given that 110 amino acid residues, out of them 40 are actual binding sites, and prediction program identified 50 binding site residues, out of which 30 are matching with experimental data. So uh, the truth set has 40 residues. The prediction program identified 50, but 30 of them are matching. So total is 110, actual positive 40, predicted positive 50. So true positive would be 30, false positive would be the difference. So 50 minus 30 will be 20. 20 are predicted to be false positives. Uh, yeah, and for false negatives will be the actual positives, which is 40, minus the true positives, which is 30. So that will be 10. So 20 and 10 are the correct answers. Okay. Sensitivity is true positive rate, by the way, and accuracy is accuracy. I'm not going to say what it is. Yeah. Wait another minute. Okay. Okay. So The, the sensitivity would be true positive rate, right? So you have true positive as 50, false positive as 15. So true positive, this is true positive, this is false positive, this is false negative, and this is true negative, right? So true negative 30, false negative 5, false positive 15, true positive 15. So sensitivity would be true positive rate, right? Which is true positive by plus false negative. 50 by 50 plus 5, 55, which is close to 0.9, 0.91, whatever. And the accuracy is basically total number of correct predictions, which would be true positive and true negative. So 50 plus 30, 80, and by the total number of predictions, which is 100. So 80 by 100, which is 80%. So 0.91 and 80. So one thing, accuracy is always given in percentages. The sensitivity, specificity, true positive, false positive rate, all are given in decimals. It's not in percentages. For accuracy, it is in percentage. So kindly remember that as well. So the first will be 0.91 and the second will be 80. Okay. Okay. Moving on to the final one. Yeah. Okay. Yeah. The answer would be blank output. There's 3 columns, and this $6 means you're asking it to print the sixth column, which is in there. So it will be a blank output. That's correct. And I think, yeah, it's the final one. Okay. We'll wait another minute, see if anyone else has answers. Okay. Yeah, the correct answer is C. There are some other, yeah, somebody mentioned some syntax errors and all. Yeah, so should be an and first of all. So the second and the fourth one are gone, and the print should be in curly brackets. So all of them have syntax errors except the third one. So yeah, that is correct. So that's end of this session. If you have any questions, do ask. Okay, if not, then I'll close this session. We'll meet tomorrow for the recap of the entire course. If any one of you have any particular topics you'd like me to cover, please let me know. Or else, like, I'll design it my way and I will cover mostly whatever has been covered in the course. Okay then, guys, I'll close this session. Thank you for joining. I'll see you tomorrow, 6 p.m. Thank you. Bye bye.